Today we will be talking about our independent research project, the effect of dissolved oxygen on yeast respiration and CO2 production. We were primarily interested in studying the biological process that relates to our own human bodies. One process that intrigued us, particularly, were the mechanisms by which our muscle cells obtain energy. Under normal conditions of oxygen, our muscle cells perform cellular respiration to produce ATP. But when oxygen levels are low, such as during hard exercise, our muscles switch from respiration to lactic acid fermentation. In particular, we were interested in the conditions under which this switch occurs. Therefore, we devoted our attention to yeast. Yeast undergo a similar switch in energy production, from respiration to alcoholic fermentation. Our intentions were such that, if we could determine the conditions under which yeast make their switch in respiration, we may be more informed about the processes of our own muscles during exercise.
Using Baker's yeast as our organism of study, our goal was to answer the question, at what concentration of oxygen does yeast switch from alcoholic fermentation to cellular respiration? In researching energy production mechanisms in the yeast, Saccharomyces cerevisiae, we found that the question not only could have implications in relation to our bodies, but is also important in the production of drinking alcohol and yeast's evolutionary history. A study performed by Dr. R.A. Wistowis and others, and an overview of S. cerevisiae by his colleague J.P. Van Dyken, revealed that compared to other species of yeast, S. cerevisiae have the unique ability of fermenting even in the complete absence of oxygen. This anaerobic quality makes Saccharomyces yeasts apt for beer and wine companies so that they don't have to regulate oxygen on the lower end. What further makes Baker's yeast an interesting organism in which to study respiration is a process called the Crabtree effect. By this effect, if there is a high enough concentration of glucose, even under aerobic conditions, the yeast will continue fermenting. A study by Arne Hagman determined that overflow metabolism, in which cells incompletely break down glucose, as well as glucose's repression of respiration, are the mechanisms of yeast's Crabtree effect. He claims that yeasts have used this process as an evolutionary advantage to starve out competitors by consuming glucose at a higher rate.
In our experiment, we hypothesized that facultative cells make the switch from fermentation to respiration even when the concentrations of oxygen are relatively low, for respiration yields a larger amount of energy per glucose molecule. When under stress of oxygen, our own muscle cells ferment anaerobically. But as soon as oxygen returns to the system, they switch back to respiration because it is more ATP efficient. Yeast, like human muscle cells, ferment even in an absence of oxygen. Yet as displayed by Louis Pasteur's Pasteur effect, as soon as oxygen is introduced into the system, yeast switch to respiration. Because respiration produces more ATP per molecule of glucose, switching would provide yeast with a more efficient energy source. Thus, switching to respiration, even at relatively low concentrations of oxygen, would provide a facultative organism an evolutionary advantage in respect to energy production, especially in conditions of controlled glucose. Based on this hypothesis, we predicted that the yeast would switch from fermentation to respiration in the first level in which we added dissolved oxygen.
As we mentioned earlier, the organism we used in our experiment was Saccharomyces cerevisiae, or commonly Baker's yeast. The choice of this yeast was both functional and practical. Yeast is one of the easiest organisms to maintain that has facultative cells. Additionally, S. cerevisiae, unlike some other forms of yeast, can decompose various types of sugars, including sucrose, the common table sugar, and it can ferment even in anaerobic conditions. Finally, Baker's yeast is available at most grocery stores and thus was easily available to us. In investigating the question at what concentration of oxygen yeast switches from fermentation to respiration, our independent variable was the initial concentration in milligrams per liter of dissolved oxygen in the solution. Our control condition was the level of no external oxygen added. We measured this amount of dissolved oxygen in 35 degrees Celsius water to be between 9 and 11 milligrams per liter, so this was our control level. Our dependent variable was the rate of CO2 produced by the yeast. The rate of CO2 production is higher during fermentation than for respiration, so we intended for the level at which we observed a significant decrease in the rate of CO2 to indicate that the yeast had switched to respiration. For each level, we started by making a stock solution of 0.1 grams per liter of sugar. We then filled a styrofoam box with 35 degrees Celsius water to act as our bath in which we would measure the yeast's CO2 production. Next, we made a stock solution of the yeast that also consisted of 0.1 grams per liter of sucrose and 0.05 grams per liter of yeast, letting that solution activate for a couple of minutes.
We then poured 25 milliliters of purely sugar solution into our measuring container, in which we would measure the dissolved oxygen using a vernier dissolved oxygen probe. We had 3 levels in total. Our control level of no oxygen added was between 9 and 11 milligrams per liter of oxygen. We measured our next level, in which we bubbled in oxygen from a tank, to have DO values of greater than 12.6 milligrams per liter. In our final level, we removed dissolved oxygen by adding 10 milliliters of 2 molar sodium sulfite solution to each trial and recorded DO concentrations from 2.1 to 2.6 milligrams per liter. To the sugar and DO solution, we then added 25 milliliters of the sugar and yeast solution. We then immediately inserted the vernier CO2 sensor into the container and let the reaction run for 3 minutes, recording CO2 readings in parts per million every 15 seconds.
One possible confound in our experiment was yeast production of alcohol, which they produce during fermentation. In too great quantities, the alcohol becomes toxic to the yeast. So to minimize the production of alcohol, we ran the trials for a relatively short amount of time, 3 minutes, and we created a new yeast culture each day of testing. Furthermore, we had to limit the amount of glucose the yeast were exposed to, for by the Crabtree effect, in high concentration of glucose, yeast will actually repress respiration. To try to prevent this effect, we kept concentration of sugar relatively low by following the examples of 2 previous labs. We also had to keep the temperature, the yeast solution, and water bath constant for each trial by using a styrofoam box as insulation so that changes in heat did not affect the rate of the reactions producing CO2. We also expected to have more levels of varying concentration of dissolved oxygen, but our DO probe could not read anything above 12.6 milligrams per liter, so we simply had 1 level for any concentrations at or above that value. While this is not a true confound, it did limit the scope of our study.
In reference to our results, this summary table shows the central tendencies, the maximum and the minimum of the rate of CO2 production in parts per million per second for our 3 conditions. The data from the 2 levels of 9 to 11 milligrams of DO and greater than 12.6 milligrams per liter of DO are almost identical in relation to all the parameters. The mean, the median, the maximum, and the minimum are all very similar. In contrast, our level of 2.1 to 2.6 milligrams per liter of dissolved oxygen had noticeably and significantly lower values for each property. These trends also held true in the t-test. Between condition 1 and 2, there was no significant difference between the means for the rate of CO2 production, meaning that the yeast in both levels was producing CO2 at the same rate despite the difference in oxygen concentration. However, the t-test comparing the mean CO2 production rates between our 3rd condition and the other 2 previous levels revealed that the yeast under the condition of 2.1 to 2.6 milligrams per liter of dissolved oxygen were producing CO2 at a significantly lower rate.
In looking at our scatter plot, the average rates of CO2 for each trial, as well as the overall average of that level as indicated by the black triangle, we observe again that the means from the 9 to 11 and the greater than 12.6 milligrams per liter oxygen levels are essentially the same. The range for these 2 levels are also pretty similar. In contrast to the near identicality between the other 2 datasets, the scatter plot really shows the significant decrease in both the mean and individual trials rate of the CO2 production in 2.1 to 2.6 milligrams per liter of dissolved oxygen.
Our line chart shows the concentration of CO2 in parts per million as it increases against time in each trial for each level. Each line represents the first 60 seconds of each trial. and the red lines represent the condition of 9 to 11 milligrams per liter of dissolved oxygen. The blue represent greater than 12.6 milligrams per liter, and the green represent 2.1 to 2.6 milligrams per liter. In this figure, we can observe there is quite a bit of overlap between the higher two levels, as the blue and red lines are pretty interwoven. However, the CO2 rates are not only much lower for the green level, but also its range barely overlaps with the other two levels.
We concluded that because we had bubbled in such a large amount of oxygen for the trials with more than 12.6 milligrams per liter of dissolved oxygen, the yeast at this level were most likely aerobically respiring. And because we observed no significant difference between that condition and the condition of 9 to 11 milligrams per liter of dissolved oxygen, the yeast of the latter level must also have been respiring aerobically. Thus, we hypothesized that the yeast in this level, 9 to 11 milligrams per liter of oxygen, our control level, were already exposed to enough natural oxygen to be respiring aerobically rather than fermenting. If this explanation is true, then we cannot make any conclusions about our hypothesis. We had originally made the hypothesis that facultative cells would switch from fermentation to cellular respiration at relatively low concentrations of oxygen. Thus, to confirm or deny our hypothesis required that there be a switch in the yeast's respiration mechanism, which we most likely did not observe.
Our results also showed that the level with 2.6 to 2.1 to 2.6 milligrams per liter of dissolved oxygen was producing CO2 at a significantly lower rate than the other two levels. Given that this was the lowest level of oxygen, we'd expected the rates of CO2 to actually be higher, for when yeast are anaerobically fermenting, they produce CO2 faster. Yet after some research, we concluded that the mechanism we used to lower the DO concentration, sodium sulfite, was actually a confounding factor. While yeast are fermenting, they release CO2 as a product of breaking down pyruvate in alcoholic fermentation. And while they are respiring aerobically, they release CO2 during the Krebs cycle. Therefore, if there is a decrease in the CO2 produced, then there must be some process either at fermentation or the Krebs cycle or before that is being inhibited. In both situations, this process being inhibited would be glycolysis, or the breaking down of glucose. Indeed, studies have shown that the presence of sulfites inhibits the function of the glyceraldehyde 3-phosphate dehydrogenase enzyme. This enzyme is responsible for the breakdown of glyceraldehyde 3-phosphate, an organic carbon molecule whose decomposition is on the path of glycolysis leading to ATP production. So, if the G3P enzyme is inhibited, then the glyceraldehyde 3-phosphate cannot be broken down, which halts the process of glycolysis. In fact, the same study we mentioned earlier found that, quote, intracellular concentration of glyceraldehyde 3-phosphate was accumulating 10 to 100-fold over the concentration in the absence of sulfite, end quote. Therefore, regardless of whether the yeast at this level were respiring aerobically or fermenting, they could not complete glycolysis. Without glycolysis, pyruvate and the CO2 later on is not produced, which explains the significant decrease in the rate of CO2 for this level.
Going forward, we would revise both our control level and the way in which we reduced the oxygen. Rather than using the natural level of dissolved oxygen in water as our control, we would probably start with a solution with the lowest amount of oxygen we could achieve to ensure that the yeast was not respiring aerobically to start. Then from this controller baseline level, we would slowly increase the oxygen amount added in small increments in an attempt to locate at what concentration the switch from fermentation to respiration indeed occurs. We would also have to change the mechanism by which we removed the oxygen from the water. To avoid any of the destructive side effects we observed with the sulfite, we would use a physical rather than chemical method. Two physical methods that have been studied to be relatively effective are purging water with N2 gas, which entails bubbling in nitrogen for an extended period of time, and boiling water in a setting of reduced pressure. To this deoxygenated solution, we would then add the yeast and whatever concentration of oxygen for that level. Thank you.
Here are references. Thank you again.